of those things during my talk. Um, so we'll have this talk, and I already Dr. Goel mentioned that you have AFM after this, right? And then there is another lecture on microbiome, microbiota, and then I will see you after that, right? So I will wrap up the day with antibiotic resistance. I think that's my plan. All right, so let's get started. Um, so we're going to talk about some of the research we are doing in my lab. So I'm going to present some of those, but I will start with probiotic. Again, the probiotic is the application. We've been talking about biofilm produced by probiotic or lactic acid bacteria, and already the day one I gave you some examples how probiotics are or lactic acid bacteria can form biofilm and their implication in industrial setting or health setup. So I just listed few lactic acid bacteria and probiotics. These are pretty well known: Bifidobacterium, Lactobacillus, Lactococcus, Enterococcus, Pediococcus, and yeast Saccharomyces, just to name few of them. But then if you look at the panel of probiotic or lactic acid bacteria we use, there are different groups. Um, so the one of the deleterious or negative effect of biofilm produced by lactic acid bacteria could be excess accumulation of metabolic byproducts. A lot of times involved in food spoilage. I had a lot of slides, actually I cut it down, I didn't want to bring a lot of information. They can form off flavor, or they could also produce some amines. You heard about amines, small amines or biogenic amines. Sometimes some of those amines could be have negative effect, sometimes more allergic effect, like histamine example. It's a breakdown product of the amino acids. Serine, cadaverine, a lot of those small amines, and it's because of the breakdown by a lot of lactic acid bacteria. Even in cheese products, sometimes you eat certain cheese and then you start showing allergic response. That's because of the breakdown of some of those proteins by bacteria to form amines. So those are the negative impact. And then of course we talked about the biofilm can be the place where pathogens can harbor or can hide, right? And in this case, even though produced by lactic acid or probiotics, that can be source as well. What about the beneficial effect? That's what we're going to talk today, right? So beneficial effect, it can have a protective effect for food fermentation, right? It can help in the food fermentation. I showed you some data last time. And also they can produce antimicrobial agents, whether it's acid or bacteriocin-like molecules. Those byproducts can inhibit pathogens. And also health attributes, right? So we're going to talk about mostly the positive, the beneficial effect of probiotics today. Again, I'm going to review some of those things you have already seen. We talked about the gut barrier, right? We went through natural or innate immune system, what we have in our gut, right? Starting with the mouth, with the saliva, stomach acid, then small intestinal antimicrobial compounds including bile, peptides, and natural microflora, and also some immune cells, right? They provide protection. We already discussed that yesterday. So that barrier can prevent pathogen from transferring through the GI tract, as well as also the lactic acid bacteria, including probiotics. They have to also overcome these innate defenses so that they could colonize in the gut and can have positive impact. What about the physical barrier? We talked about that again, right? Let's review it briefly. So we are looking at the gastrointestinal barrier, and we are talking about the physical lining of epithelial cells. So these are the villi structure in the gut, and if you look closely, you have seen already these are the villi structure, right? And these are the crypt, and these white locations are the goblet cells secreting mucus, right? And this is kind of an example of healthy ileal villi structure, and these you will see also the mucus, okay? And these are the crypt where the epithelial cells origin, and they migrate upwards as the cell mature and die, okay? And if you look closely, then you can see this epithelial cell lining. If you take two epithelial cells, and here is one, and this is another one, and this is the cell. And this tiny junction is called tight junction, right? This is the tight junction between two cells. And this is the microvilli structure, filamentous structure. Now go deeper into the cellular architecture, then you will see this area only. And if you magnify it further, you can see this little area. These are the microvilli. This is called tight junction. Going over that, this is adherence junction. This is desmosome. This is gap junction. This is how the epithelial cell line up, right? And if you look closely, what are those proteins? We went over that a little bit. You see these tight junction proteins are claudin, occludin, zonocludin, F-actin, and then the myosin light chain kinase. These are the components of the tight junction, right? And then the adherent junction, the major protein is E-cadherin. Then also you have alpha catenin, beta catenin. These are the proteins holding this junction together, right? And then you have also seen this, the schematics, how these proteins are held together in the tight junction or adherent junction as well as the desmosome. Then as I mentioned that also you can stain individual proteins to assess the epithelial integrity by measuring the protein level. So this is confocal imaging showing you the presence of zona occludin, and these are the E-cadherin, which are the long proteins. If these E-cadherin proteins are destabilized or broken down or degraded, you will not be able to see this protein, right? This disappears. And this is the way we can monitor the integrity of the junction, epithelial tight junction. What else I need you to know? If we talked about few times about the leaky gut, right? The epithelial permeability, if it enhances due to infection or due to heat, due to any abnormal conditions, you experience leaky gut, right? And the level of the pore formation, literally you can measure the level, how the permeability occurs. So if it is a pore pathway, which is very small, about 6 to 8 angstrom, that means the small molecules can pass through from the lumen to the subcellular spaces. If the epithelial barrier disruption is about 100 angstrom, and we call it leak pathway. And that's when the water and the other molecules start to leak out. And the best way to assess that, you can measure the flow of two different types of molecules. And FD4 implies actually fluorescent FITC conjugated dextran molecule, about 4 kilodalton, and this is 40 kilodalton size molecule. So what we do, we measure the transfer of molecules from the upper layer in the transwell setup when you grow the epithelial lining, and then you look for that molecule in the basal layer, right? If there is a tight junction compromise, this small molecule would go through. And based on the size of the molecule passes through, you can calculate the pore size or the level of disruption or barrier damage. And on the transwell setup cell culture model, you can definitely calculate that, as well as bacterial translocation. And the unrestricted pathway is the one when your tight junction is totally wiped out. Means sometimes the damage happens so severe, the bacteria can easily pass through. So the larger molecules, bacteria, they all pass through. So if you have a diarrheal situation like Shigella dysentery or Salmonella or Campylobacter or even Helicobacter, or Vibrio, sometimes they cause severe damage. The bacteria also can pass through. So these are the level of epithelial barrier damage or dysfunction you can measure. And in the animal model, how do you assess it? We need to feed the animal about 4 to 5 hours before you sacrifice it. Okay? Because a lot of our data I will show you, we measure that in the mouse, and you orally gavage them 4 to 5 hours before sacrifice. And most of the time we use FD4 because you try to measure the leak pathway. That's where we are interested. But you can measure also FD40 or FD70. These are the larger molecule. And if they pass through, that means you can assess your damage is severe. So that means depending on the agent, whether it's a toxin or any abnormal conditions in the gut, you can literally measure the level of barrier disruption you have. Okay? That's very important, because if you have a severe damage, then repair would take longer, right? Because we always want to repair our gut barrier dysfunction so that you can heal fast, and that's the part of the gut health. And so after we sacrifice, we collect this serum and also urine, and we monitor this molecule in this fluid, whether it's in the serum or urine. So if you see high level of this molecule in serum or urine, then you know you got a severe gut problem. Okay. In human also, if you go and visit gastroenterologist, they will give you some drink and let you drink that. After that, you can collect it in the serum, mostly in the urine. They will measure it, and if you have a high level, that means you got a gut problem. Okay. All right. So this is how we're going to use it in our experimental approach. And a little bit more, I want to tell you. And if this damage happens in the tight junction, and this is also called paracellular migration. So that means microorganism, they can migrate through paracellular route from lumen to basolateral side, in the below the epithelial cell, and then it gets to the blood circulation. Okay. Some microbes actually they produce enzyme, and this enzyme also can break down this protein, and so it opens up the path. Okay. And some cases the organism can activate actin skeletal, means the cellular protein structure which maintains the integrity of those cells, and if you disrupt that actin molecule, and sometimes this junction open up. So these are the varieties of ways microorganisms manipulate the cellular function so that they can get inside. Okay. Again, look at it from the pathogen side. This is what we are looking at. This is another way. So one of the, again, you saw this picture before. What is the signaling cascade that leads to opening of this tight junction? Okay. Sometimes, like we talked about LPS, peptidoglycan, right? Those gut-derived microbial components, and they are highly inflammatory, right? They can also cause tight junction barrier dysfunction. And what they do, if they are binding to a receptor, and then this MyD88 pathway, they can activate NF-kappa B. Okay, and we talked about that NF-kappa B signaling cascade, and this P55 or P65, they're all actually in a normal state. They're forming a cluster, and when they're activated, they separate and they get inside the nucleus. The first thing they will do, they will bind to the promoter, and then they will transcribe this, and then from messenger RNA, and the result is, outcome is inflammatory cytokine, like TNF alpha, IL-1, IL-6. These are the inflammatory cytokines are produced. So that's the indication that inflammation is taking place. And this low level of cytokine we talked yesterday, it's good for your health from the standpoint of activating immune cells. Okay, and if this happens severe, and then it come back also, it can also regulate this tight junction barrier. So it also make it leaky gut. Okay, and one of the other protein I mentioned is that myosin light chain kinase. And this kinase enzyme controls this tight junction protein. So that means this tight junction protein is holding together. If the enzyme get activated, these proteins are pulled from the junction area. Are you following me? So these proteins which are holding together, I showed you before, and this tight junction is kind of like a sewing, right, with the thread, and this enzyme is get phosphorylated, MLC2 phosphorylated myosin light chain, and then actually it controls these proteins which are holding the junction together. So if I'm a microbe, or if I'm the stimulant, I can activate this enzyme, and then what it does, it pulls all the proteins away from the junction and open the gut barrier. The epithelial become leaky. Are you following me? So that's when the molecule from the lumen can get in, or the molecule fluid from inside can get out. So that's the during diarrheal condition. Okay. So this is one of those established mechanisms used by at least some of these molecules. And this is just showing that again in the cartoon form, showing that these proteins, how they are regulated, and they can pulled away. And usually we call it, these proteins recycle, means it's gone inside the cytoplasm, and then few minutes later it comes back. So it's not a irreversible process, it's a reversible process. You can make it irreversible if it is a severe damage happens. It takes longer to repair it. Okay. So this is normally continuously happening in the body. So if you are heat stress, okay, this can open up. That's when you show signs of diarrhea or vomiting. Okay. So that's example. If you want to learn more about it, the several different microorganisms, we wrote a review years ago, and you can see all those enteric pathogens listed here. They use certain virulence factor, and they also target this lot of these proteins or signaling pathway to open the tight junction barrier. So that's how they get easy access to our intestinal mucosal layer and also distant organs. Okay, and we also mentioned about the leaky gut, right? Look at this picture. You saw this before, right? Whether it's a pathogens or any natural organisms in the colon, colonic tissues, or in the gut, they can cause inflammation, right? Inflamed epithelial barrier damage. Also colonization of opportunistic pathogens. Here's a Staph aureus. Microbial dysbiosis, right? All of those conditions can cause leaky gut. So therefore, this microorganism is going to come below the epithelial lining and then activate immune cells. As I said before, that you don't want excessive activation of immune cells, and they cause cytokine production, exacerbate the inflammatory process. Okay, and then also we talked about the consequence of having leaky gut, right? And also all those molecules getting in, then all the cytokines and the molecules circulating in the body. So whether from lungs or gut, you end up getting into the brain, right? And also the joint. We talked about arthritis, right? That's where the antigen antibody complex or some of the cytokines accumulates, cause inflammation or swelling. And then also the gut brain axis. Okay, so as I also said before, that leaky gut leads to number of different diseases. So it's a serious consequence or consequential disease form. So allergic or autoimmune conditions, asthma, atopic dermatitis, allergic rhinitis, celiac disease, inflammatory bowel disease. So a lot of those diseases linked to leaky gut. Also I mentioned that like diabetes, obesity, rheumatoid arthritis, multiple sclerosis, autoimmune hepatitis. So there are a number of diseases linked to leaky gut. Alzheimer's, Parkinson's, chronic depression, autism. So a lot of the diseases are now may have started from the gut. Okay, so leaky gut is very important. So how do we repair all of those things? So that's where I'm going to talk about the probiotic and what we did in our research to help mitigate some of those gut related complications that we repair the leaky gut. I don't need to spend time on defining what is probiotics, prebiotics, symbiotics. I presume all of you know it. I just want to point out only a few things about probiotics. So there are some consensus people, the experts, they met and they are trying to redefine what is actually probiotic. Sometimes it gets really confused. So if you see any lactic acid bacteria, they use for fermentation of product. Do you call them probiotic? By definition, no. One way you can call it probiotic as long as you can demonstrate that it has a health beneficial effect. Okay, so that's the key word, health benefit. Okay, if they don't have a health benefit, direct health benefit, not indirect one, and then you cannot really call them probiotic. Other aspect is that you know this massive microbiome, microbiota research we are doing, right? The whole purpose of this is that if you know or not, why we are studying microbiota, microbial community. At the end of the day, we want some microorganisms, we can use them as probiotics. Okay, if you can identify whether it's for skin disease or heart disease or gut ailment, if we could find or identify some organisms from this unknown community, we want to use them as a probiotic eventually. Okay, and that's where we are heading. And so this panel decided that can you call this microbial community as probiotic? Can you call this microbial community you are now studying and you are saying that this number of organisms have certain impact on your health? Okay. The question is, can you call them probiotic? Because a lot of times people may claim that, oh, I got this from this community and I can prove that these are probiotic. Can you claim them as a probiotic? So the answer is, I would say no, unless you define those cultures clearly. Genetic information as to genetic, what taxa they are, what their function, every detail of them. Unless you can do that, then you don't have any claim to establish. So they are trying to warn you that if you have a microbiota transplant, fecal microbiota transplant, right, you cannot call them probiotics because it changes. You have no clue what are the population, you have general idea, but you may not know every detail of those organisms. Okay, like we know about lactobacillus acidophilus, lactobacillus rhamnosus, or bifidobacterium. Those are known probiotics. So that's the warning they're trying to give you. So keep that in mind as you study. Then also we use a lot of definition for probiotic, right? So not only probiotic, postbiotic, pharmabiotics, next generation probiotic, all kinds of names there. I want you to again familiar with that, okay? Where we are going with this probiotic research. And then also there are some known organisms I already mentioned. Some of those unusual ones like E. coli, they use that Miscellaneous strain as a probiotic. And then spore forming bacteria like bacillus, they are also very good probiotic. Do you have any idea why people like to use spore formers? Spore former probiotic? Any idea? Spore formers, they can withstand heat. They can withstand a lot of processing conditions, right? So that's why it's easy to use it in a product because they have a self-stable, self-life, right? If you are formulating a feed, and that can have a lot of temperature abuse, right? If you have live organism as a supplement, they may die. But if you have a spore, they will survive. And as soon as they get into your GI tract, they can become vegetative cell. Now they can exert their probiotic effect. So that's why a lot of company, they like that the spore former for practical application. Sometimes even some people try to add it to a cake mix. Think about it. Cake mix, they will add spore former. And so during baking, with the idea that they are going to convert into vegetative cells. And that's when they can have a probiotic action. If you use just the regular lactobacillus or other vegetative organism, they are not going to survive. So these are some of the clues, remember those. And, this is in US, okay. One night I sat down in the computer and tried to find out how many different probiotics are available. It's huge, enormous. It is growing now. I mean, I know it's in the Eastern culture, people here in the Asian countries, people use a lot of probiotics, right? And there are a lot of traditional use from generation after generation. In the Western culture, especially in the North America, this is beginning to gain momentum. People started to believing probiotics has positive effect, right? They never believed that probiotic has any good effect. So now once they understood, now a lot of companies try to make money out of it, right? So they will market all of those, a lot of these are similar organism, maybe same organism. They use multiple strains of same organism. It's kind of ridiculous how they market it and they sell it. And you go to pharmacy or grocery store or any health stores, you'll find a lot of these probiotics in the shelf. So I think I kind of raised questions that do they work? A lot of those questions I will try to address that. But a lot of times this is a feel good, right? A probiotic, it has a positive effect and they sell. And also for the pets, right? Pets also is a huge market. The cats, dogs, that's very big. And of course the livestock also. But these are all actually for humans. And of course the symbiotic, I don't want to again spend time. There was a review by the consensus panel. They talked about symbiotic. They came up with the certain criteria to establish a symbiotic. Just the two definition I want to point it out. Complementary symbiotic. So the two symbiotic with the probiotic, if you mix it, they complement each other. They don't compete. And the other one is called synergistic. So like if you have a symbiotic, probiotic, the action should be added. You see enhanced effect. We are doing one research with our probiotics with blueberry polyphenol. We noticed that if the probiotic has one effect, the polyphenol has effect, and if you add two of them together, you see right here. And that's a true example of synergistic effect. Okay, just to point out, again, those the references are there to follow. This actually is this most important slide, or there's another one I will talk about. I tried to cover a lot of these things yesterday. Due to lack of time, I didn't get enough time to talk about the immune response, right? So we talked about food antigen, right? You eat food naturally and how your body responds to it, what immune system does. And we discussed also that we don't develop immunity against food, right? Then we will not survive. Food is a foreign antigen, right? And if you have immune system against food, it's going to be a problem, right? But we want an immune system against pathogen. So just to, I don't know whether we'll be able to focus so much on this left side, food antigen, right? So we have a healthy condition, mucus, antimicrobial peptide, and then IgA, right? And then what you look at this immune response, okay, it's fascinating that all those this TGA beta, IL-10, IL-22, a lot of these are upregulated. And the type of cells, immune cells, I started talking a little bit, regulatory T cell. This is also another one, Rho gamma T. This is also T lymphocytes, macrophage B cells, and CD103. This is actually dendritic cells. They have a positive impact, okay? And the antigen processing we talked about, right? Antigen presentation, and they have a greater tolerance for it. They don't present to the immune cells. So therefore, the immune cells don't, or T cells, don't think that they are foreign. So that's why they are not going to induce immune system. And if you look at the mucus goes up, AMP up, IgA up, PR pattern recognition receptors are up. But if you look at the pathogen, the dysbiotic condition, the disease condition, right? Those are all negative. See this going down here. Look at the type of cytokines goes up. These are all inflammatory cytokines. And these are the clear marker. The type of T cells also respond. These are clear marker that you are under dysbiotic conditions where your immune system is very weak, very poor. They are showing inflammatory response. Do you see the balance? And this balance always playing a role how when you take food or probiotic or healthy diet, okay, healthy conditions, versus if you have a dysbiotic or poor food habit, like a Western diet. We talk about hamburger, pizza, all kinds of nasty, the sugary food, all kinds of stuff have fall under this category. So all negative effects. And the healthy diet is fall in this side. And so the immune system works very positively in that side. So keep that picture in your head. And the other one, again, is pretty complex. I would recommend you to go through this article published in Nature Medicine. And this essentially summarizes how some of those probiotics positively impact health. Okay? If I can just quickly tell you that they produce short chain fatty acids, and they also the forming biofilm is one of the criteria. This is the community. And remember, I talked to you about that that can slightly cause inflammatory response through LTA. I think we had a discussion yesterday, and also with the shortage dependent pili we had talked about yesterday. So they can also activate these epithelial cells. Okay, and then also the epithelial dendritic cells bring those antigens or probiotics inside, and it also initiate this regulatory T cell pathway, which are the immunomodulatory response. And there also you will see the anti-inflammatory response, and also the tight junction. The probiotic, it maintains the tight junction. It works opposite to pathogens. If pathogens open it, probiotics will close it. And this is again, you see upregulation of claudin, zona occludin protein, mucin, MUC2 I mentioned, their level also goes up. So these are all positive attributes due to probiotic. And also GABA. I think some of you are working GABA and some of these probiotics, and these are also secreted, and it also directly affects the brain. Okay, and then also the other functions. So if you go through this review, I think it did a great job summarizing those immunomodulatory, anti-inflammatory, GABA, all of this response, right? So let me show some of our data. Okay, and I think I set up the base, the background well, so you can follow this research. And so this is a company, small company, gave us some money, and they market for probiotic blend for their pet food. They have a dog feed they sell, and they gave us this probiotic to do some testing, asking us how well this works. And so Nick, my grad student, he started studying a little bit on that one. So what we did, this blend actually has these three organisms, pretty recognized organism, right? Enterococcus, lactobacillus, lactic acidibacillus, KGI. And so few things we did, very simple study, just to show you what properties we are looking at. Because it is for dogs, so we had to use canine cell model, which is the kidney cell. And here we looked at the pathogen inactivation, biofilm formation. As you recognize, biofilm characteristic is one of the important characteristics for probiotics to have. We recognized that a while ago, and many people now recognize that. And then also we looked at anti-inflammatory response. And first, I think it's very simple. You measure the activity, right? If they work against some of those pathogens, common pathogens. So what you noticed actually, the bacteria has that antimicrobial effect, but not the supernatant. If you don't see the activity in the supernatant, clearly says that probably these bacteriocin-like molecules are not quite involved. So we haven't really want to explore what is causing it. That was not the purpose. We just wanted to screen with a limited amount of funding, so we just left it there. But most importantly, we wanted to see, do they form biofilm? If we recognize biofilm formation is one of the positive traits or attributes, so we want to measure that. So we compared that with Staph aureus. In 48 hours, you can see Staph aureus form a strong biofilm. But whereas my lab max 3 did not quite form that, and LKGI also pretty weak biofilm. But after 72 hours, Staph aureus remained pretty same or similar, but these two other ones picked it up. So that means it's time-dependent biofilm formation. They take longer, okay? And just to tell you that how long, make sure the animals are exposed or how often you have to feed them so that it's maintained certain level, right? In order to get the positive attribute. And by counting colonies, this is by crystal violet staining. By colony counting also you can put the same information. And then you can also stain it by Giemsa stain. You take the biofilm, and here you can see this the bacterial biofilm by these three organisms, and you can see clearly the strong biofilm formation. Another thing I also mentioned that the morphology may change, right, in the biofilm producing cells, and we did indeed notice the changes in the cell morphology in those organisms. Anyway, and then of course we wanted to see the adhesion to the kidney cell line, and you can see the LabMax 3. What we did actually in this case, we treated with LPS just to make the cell simulate the gut environment where your epithelial cells would be exposed to LPS, peptidoglycan, a lot of those molecules coming out of the bacteria. And so we wanted to see how well this works. And so naturally, if you look at all of those data together, you see this LabMax 3 form nice, impactful colonization on the MDCK cell line, which is the canine cell line, and compared to KGI produced a little bit more. And so they adhere, they form cluster like biofilm-like structure. And they are also non-toxic. And by LDH assay, you can confirm that these cells are not toxic. These probiotics are not causing cell damage. Okay, and then of course we looked at the cytokine profile. Okay, and the cytokine profiling, and definitely one of the desirable cytokine we wanted is the IL-10. But the most importantly, the ratio between IL-10, which is anti-inflammatory, and TNF-alpha, which is inflammatory. So you looked at this, the ratio is pretty low, so indicating that LabMax 3 is working moderately well, as could be a positive one like a lactobacillus KGI. Anyway, so we had some indication that it does immunomodulatory or anti-inflammatory response. Okay, so now I wanted to get into the what bioengineering strategy we took to help improve these probiotics to work against some diseases. Okay, so in this case, we wanted to see why some probiotics don't work. Okay, I'm trying to ask this basic question. Like a lot of times you take probiotics, it works. Sometimes it doesn't show positive effect, right? And that's where we are concerned about. So let's ask this question. So the probiotic organisms, you'll find them embedded in mucus layer. Okay, they never enclose to epithelial cells. This is under healthy condition. What happens if you are under disease condition or dysbiotic condition? So what dysbiotic condition looks like, your mucus layer thins out. We saw that, right? And then epithelial barrier also disrupted, right? And in that conditions, this is when you want your lactobacillus to be around, right? If they are probiotic, but they are the one which leave the body. So if they are not there, because the condition is not favorable, and they will disappear, therefore you will not have any positive effect, right? And that's the problem, right? So if you have leaky gut, we talked about inflammatory diseases, if it is in the animals, poor health, weight loss, disease susceptibility, survivability, and economic loss if it's for the livestock or farm animal. Okay, For human, we already know what consequences. So the question is, can you reverse this process? Okay, and reversing the process requires some diligence from your side, the scientific discovery. So one of the things we are looking at, what is the solution, right? And so the question is, what happens during dysbiotic condition? What do we know about the dysbiosis? Why these lactobacillus are not staying in the gut when they should be, right? What can we do to keep them? So one of the things we noticed that heat shock protein 60 is one of the molecules that expressed at high level in the disease cells. Everybody with me? Okay, so that's one of those markers we are trying to work on. So what is HSP60? Okay, I'm going to explore a little bit more. So during infectious disease, it increases. Also inflammatory disease, metastatic cancer, cardiovascular disease, this molecule goes up. This is one of the greatest indicators. We are exploring some. This is not much data available. We are doing some work to understand why this molecule goes up. So we have a protein that binds to HSP60, okay, and that protein is called Listeria adhesion protein, or ADHE, which is called acetaldehyde alcohol dehydrogenase. Okay, this is one of those enzyme or protein. It's secreted by many organisms. Okay, and so what we wanted to do, if you can create a probiotic, are you following me? This is where the most important part coming in. We want to express this probiotic organism with this lab, okay? And this lab is coming from Listeria. This is where we worked on in our last 20, 30 years. And so we figured out, if we could express lab which reacts on HSP60, can we have this probiotic have a strong binding to those epithelial cells which are under stress, which are showing the signs of disease condition. And this is what we wanted to do. Okay, so HSP60 briefly, if you don't know about it, it's called chaperone protein. If you know, chaperone is that it deals with the protein synthesis, protein misfolding. They will remove those things from the cell, and also they have an immune lighting function. Some people claim, or we also claim, that immune signaling molecule, and it's a disease marker. Already I mentioned that. And so if you look at the cancer cells, they are finding out the HSP60 playing an important role for any kind of metastasis. What we found out, actually, I mentioned about TLR molecule. This HSP60 likely binds to TLR4. It activates NF-kappa B, and so it controls a lot of aspects of the cellular physiology, even the cancer. Okay, and also in the cardiovascular disease, what we are finding out is that if you have a hypoxia condition, like a lack of oxygen, and you are under stress, so HSP60 would be produced at high level, and some of those HSP60 is secreted, and also it can bind back to TLR4 I mentioned a minute ago, and it binds, it activates this inflammatory pathway, NF-kappa B pathway. Then you see a lot of inflammatory cytokines, and that complicates your heart disease or heart attack. Okay, so these are like another mechanism to figure out what happens when you suffer from cardiovascular disease. But this is one interesting aspect. I just pointed out that HSP60 expressed on the membrane to serve as a marker or serve as a signal that you are under stress or you are under bad situation, and then it could be health risk. Anyway, so we are interested in Listeria monocytogenes pathogenesis in our lab, and we said that, well, under Listeria monocytogenes infection, HSP60 expression goes up, and so naturally in your gut you are going to see some HSP60. Okay, and you can clearly see this HSP60 expressed on the epithelial cell outside. And my student many years ago, she actually, Jennifer, she first reported that lab binds to HSP60. So HSP60 is the receptor. And then later Christine, she reported that under stressful condition, including Listeria infection, you see a high level of HSP60 expression. And later on, we also proved that in another research. So we know HSP60 play an important role in Listeria infection, or Listeria literally can bind to HSP60 during infection. So I'm going to tell you a little bit. And I already talked about Listeria. I'm not going to spend much time on that. It's a foodborne pathogen. I also talked about they can cause gastroenteritis, neurolisteriosis, or pregnant women complications, right? And so we know that it's a foodborne pathogen. It comes through food. It passes through the GI tract. It can get into the mesenteric lymph node, blood circulation, then eventually brain, and in the mother, it can cause the fetoplacental barrier, right? And the fetus doesn't have immune system. Therefore, it cannot defend against listeria. And so the fetus are highly susceptible. So stillbirth, premature birth, fetal death is very common in listeriosis cases, and it happens number of times. In US, it's always like anytime you have listeriosis outbreak, some women get their problem with the fetal death or premature birth. So it's highly problematic in humans. Of course, in animals also it can cause disease. Originally, it came from cow. It caused circling disease because it affects the brain, and sheep also have abortion. Okay, so it's a true example of zoonotic disease. Okay, but it's a human pathogen. So as I said, it has a lot of pathogenic factors to work with. But our research lab, we contributed to the understanding of Listeria pathogenesis. We discovered that Listeria adhesion protein, that's we discovered, which is alcohol acetaldehyde dehydrogenase. It binds to HSP60 and it activates signaling cascade that leads to infection. Okay, so just briefly, in order to understand the bioengineered probiotic, I want you to understand a little bit about this lab protein, which is acetaldehyde dehydrogenase, alcohol dehydrogenase, right? And this is part of actually glycolytic pathway, glycolytic pathway. Glucose converted into acetyl-CoA. All of you should know, right? Glycolytic pathway. And then acetyl-CoA under aerobic condition, it can go to TCA cycle, right? Again ATP, right? And if it is anaerobic condition, bacteria can harvest some energy, and which is ALDH can convert it to acetaldehyde, and alcohol dehydrogenase convert it into ethanol. Okay, so this enzyme is called bifunctional enzyme, and it's about 96 kilodalton. We call it moonlight protein or housekeeping metabolic enzyme. It has a dual function: enzymatic function when it's need, harvest energy. Other function is pathogenesis. Okay, that's the beauty of it. That's why you call it moonlight. You don't see moonlight all the time, right? In a month. So you have a cycle, right? And so that's why they once in a while they serve as a pathogenic factor. And other interesting aspect we learned, you find it in the pathogenic and non-pathogenic bacteria. And non-pathogenic means Listeria inaqua example. Agree with me? So why non-pathogenic using this protein, which is a pathogenic factor? So in non-pathogen, it doesn't have that attachment function. Okay, only in the pathogens, and it has a strong binding affinity to the HSP60. So what we learned actually, this is Kwang Pyo and Bala. They did quite a bit of work. Actually, Kwang Pyo did. He's now professor in Korea. He's doing quite a bit of work on biofilm research. Bala is working for Nestle. So they did some initial work. So one of the things I mentioned that this protein doesn't have a leader sequence. These are inside the cell. And I showed you how they are excreted outside before, right? So they, in this case, this is gram positive. They is called SEK pathway. I mentioned that SEK2. This protein, it binds to the SEK-YEG and then gets out. And then if it is outside, and if it is not bound to the surface, it doesn't have any function. So what happens in pathogens, it binds to a receptor, which actually Dong Si proved that, and he published that recently, last 23. And he said that this protein gets out and it finds a protein to lodge, whereas the non-pathogen, they don't have that receptor, and therefore it gets lost. So both pathogen and non-pathogen make this protein. Are you all with me? Listeria monocytogenes is pathogen, it makes this protein. Listeria inaqua, non-pathogen, it also makes this protein. But inaqua, it doesn't have this anchor. Okay? Therefore, this protein gets lost. But this protein is identical, only except I think I would say 3 or 4 amino acids out of 866 amino acids. Anyway, those proteins are same. So we also recently tried to understand the lab HSP60 complex. We finished that work, it's submitted, and this is a cryo-EM. You heard about a little bit about yesterday, the structure part. Here we did cryo-EM to understand the lab protein, and it comes as a pentamer, tetramer, and also it acts as also dimer. It comes with varieties of structure. It's very fascinating. Anyway, no time to discuss more detail, but you can see this. This is HSP60. You can see this one, multi-dimer, and this small part of the lab actually binds to it. Okay, this is what we understood. But without spending much time, this is just the illustration showing the lab binds to this receptor, which is called it internalin B on Listeria cell surface, and then part of that lab binds to HSP60, which is host cell. Are you getting it? Listeria binds to this host cells before it causes the damage. And so that's the mechanism which already revealed and we submitted it, is under review right now. So how does lab help in the infection process? Okay, you remember this picture? I showed you this one earlier, right? Yesterday or day before. And so here actually lab binds, it activates NF-kappa B pathway, it opens up the tight junction barrier, and it passes in between. Okay, and then because it is an intracellular pathogen, this bacteria also can invade through cell by another pathway. Okay, and this is what we learned, because activation of myosin light chain kinase removes the claudin, occludin, and ecadherin, and that's how it goes through. The other aspect is the part of the innate immune system, and so before immune system wakes up, it can infect the host cell. Okay, and so quickly, other aspect also you learn that it uses called caveolin mediated endocytosis. Again, we use the knockout mice to prove that caveolin also help in the translocation of the bacteria. Not have enough time to go through this cellular mechanism, but just to get the big picture. Okay, the cell junction opens up, bacteria pass through. What are the cell biology? Which proteins are involved in opening this junction, right? Because you have to understand, if you can close it during the disease or the repair process or the healthy gut process, right? So that's where we are going to get in. This is just saying, again, understanding cellular biology, how this caveolin form a complex with the occludin, and this complex with the Listeria monocytogenes. This is a clear evidence. They form this called caveolar endocytosis pathway to go through the cell. Okay, all right. So this is the summary picture. Again, if you are interested, this was published last year in MBio. Rishi was taking the lead in this research. So can probiotics prevent listeriosis? That's the first question we asked. That's what we were waiting for. So one of the things we did actually, that's Oki and Ku actually, she worked on it, and this is the first time we ever worked on probiotics. We had no intention of working on probiotics ever. We were working on Listeria pathogenesis. But in order to prevent Listeria, all this knowledge we gained, can you use probiotics to prevent Listeria infection? That's when we got into probiotic research. So you can say our knowledge goes back to 12 to 15 years maximum. Okay. So we looked at Listeria monocytogenes. We used just the traditional lactobacillus, rhamnosus, acidophilus, paracasiae. None of them actually inhibited Listeria on a cell culture model. Okay. And I already said that before. Sometimes Listeria probiotic works, sometimes it doesn't. So we want to make it work. So what we did actually, we engineered this probiotic. And Rishi, actually Marianne took the lead, and then Rishi completed it. So they were co-authors. So just briefly explain to you what we did. So we took that lactobacillus KGI, right? And we took this lab from monocytogenes and Listeria inacua. One is pathogen, one is non-pathogen. And I mentioned before, these proteins are same, sequence-wise. So why not take it from both? Let's see if they can protect. So once you create it, so you express this protein with this expression system. And these are two bioengineered probiotics. Sometimes I call it BLP, bioengineered lactobacillus probiotic, or we call it NGP, next generation probiotic. So I have used this interchangeably. And so first thing you want to prove that these proteins are expressed on the surface. They did. Okay, so you can clearly see that. Whereas the vector or wild type, they don't have this protein expression. Okay, first thing you want to see is that do they attach to the epithelial cells, as you promised. Okay, so if you look at the wild type or vector, they have a minimal adhesion to KCO2 cells. But if you have a BLP, they have a higher adhesion, because this is the adhesion protein in Listeria. And do they invade? They don't invade the cells. That's a good news, right? Do they go through the paracellular route, translocation route? They don't. So that is very good information. Then next we look at, is it HSP60 specific? So one way to do that is that we created a KCO2 cell line which has the HSP60 knocked down and also overexpressing. So if there is a standard HSP60 expression, this bioengineered probiotics so increased, adhesion and exclusion of Listeria monocytogenes. And in this case, overexpression, you have a higher binding, no HSP60 expression, no binding. So that's the HSP60 specific response. And then we wanted to prove that in the animals. See how well it worked, right? So we used mice and we used pregnant guinea pig. And so whole idea being is that we fed them, this is more like a prophylactic. So you take the probiotic. That's what we do, right? You eat it for 10 days. And then we challenge them with the pathogen orally. And then we kill the animals and then look for the Listeria in different organs and tissues, as I mentioned before, right? And we did that in mouse, normal, and this is pregnant guinea pig because pregnancy is a high risk factor, right? So let's look at the video. So this is naive LM control. This animal extremely sick. It's 48 hours later, right? This animal is not moving. It's going to die pretty soon. This one received wild type probiotic, right? And they are challenged with LM, and you can see equally sensitive. It's also terminal stage. Let's look at these two on the right. One receiving probiotic made by LM lab, and this is Listeria inaqua lab. Are you with me so far? Okay, now let's look at that animal. That's quite healthy, right? It's moving around. What about this one? This is even more active. The whole idea being is that this bioengineer we created, we challenged them with LM, now they can resist the infection. And when you look at the number, actually LD50, I mentioned lethal dose 50, we are getting about 92% protection. The high level of animals are protected from this infection. So now you may ask the question, how, why, what is the mechanism, right? We address all of those things. If you want to read that paper, it got tons of data. And so they have increased adhesion, colonization in the ileum, cecum, colon, all those bioengineered probiotics. The most importantly, we wanted to see, do they form biofilm? That's important, because how do the mechanism work? So I don't know if the lights are dim enough. So here actually wild type, they are mostly present in the mucus. They never goes close to the epithelial cell. Whereas the bioengineered one, they attach directly to the epithelial cell, both of them, and clearly show that they are binding to the epithelial cell. It's called epithelial cell contact. And that's very important, why this works. And if you count them, you can clearly see there are high number of those probiotics attached to this epithelial cell. Do they form biofilm? Next question. And this one clearly shows, you can see this, all this biofilm, including mucus, sitting right on the epithelial cell. You will see that wild type, because we don't have the right marker to treat it, so we don't know. And so here also is showing that there is a competition between Listeria monocytogenes and bioengineered probiotics when they are binding to the epithelial cell. There are some elegant experiments we did. No time to discuss that, but if you want to know more about it, I'll be happy to talk offline. So they maintain epithelial barrier integrity. We showed that, right? Do they do that? So again, use the cell culture model. If you treat with the Listeria monocytogenes, this opens up. The molecule goes through, or Listeria goes through. If you have a bioengineered probiotic, look at the FD4 movement. It decreases significantly. So if you have a leaky gut, now with the probiotic, you can prevent the leaky gut. You can repair it. What about in the mouse? You can see the exactly same result. So here is the LM showing the FD4 leakage in the both serum and urine. And now with the bioengineered probiotic, you can see already like a control. So that improved also in the urine. So that's saying that it does. What about the protein lab? I mentioned about those proteins, right? So I would set it up, myosin light chain kinase, PMLC, claudin, occludin, ecadherin. Look at those proteins. Look at the LM. LM causes severe damage. All those proteins like claudin, look at this, disrupted. This is control to follow this. Occludin, also you can see they are severely disrupted because of the LM. Ecadherin, also these long proteins all disappear, disintegrated. So that's saying there is this barrier damage. What about this probiotic now? So even wild type did not do anything. But when you have these two, you can clearly see this claudin restored and ecadherin restored, and all those other proteins, whatever it's supposed to be. So it's kind of reversing the whole process, only because they are attaching each other. Okay, so that worked very well. What about the cytokine? inflammatory response, anti-inflammatory, TNF alpha low, IL-6 low. What about the regulatory T cells, which modulate your immune system? I mentioned that earlier, right? Treg cells. And they are, whether you have probiotic or not, is working very well. And if you have a Listeria monocytogenes infection, then also it works exactly the same way. And this is a good sign that you are modulating immune system to a level where you can improve the health of this animal. Okay. What about pregnant guinea pig? Let's look at that. If it can prevent pregnancy-related complications, this is very important. So Valerie was involved in this project, and this is a pregnant guinea pig. And I don't know whether you can afford to do this experiment in a routine basis. Guess what guinea pig cost? This pregnant guinea pig. We bought several of them. So each of them cost anywhere $300 to $400. So calculate that in rupees. It's about 85 rupees per dollar. Okay. So to do this experiment, you really have to have a lot of money. So at least we spent $15,000 to buy just the guinea pig. So you have a statistically equivalent number to prove it. Anyway, let's see if this probiotic, these guinea pigs were fed with this probiotic. And literally they like it. We have to give them in the water. They will just go, and instead of drinking water, they will just drink the probiotic. And we don't want to physically abuse any of those animals, say force feed them. We don't do that. Just leave it there, they will drink at their pleasure. And so they've been doing that. Then we challenged them with Listeria. Now, one thing we did, we wanted to do subclinical infection. Very low doses of infection, because we don't want to take a risk of getting abortion. Then we don't get the result. And so we did a low level of infection. So the mothers, if it happens, if you are susceptible to listeriosis, sometimes you may not show any symptom, but the fetus would show symptoms. Okay, so it's like a asymptomatic disease in pregnant women. So we try to prove that. And so when you did the low infection, mother is actually infected. If you look at the liver, mesenteric lymph node, all of them were positive. Spleen positive, liver positive. So most of the mothers, they are all positive. Blood also, we see less. That's good news. Lungs also, this bioengineered fed guinea pig are less, almost zero. Right? Kidney also zero. Whereas the wild type did not have protection. Let's prove that in the placenta. What happens? This is where actually magic happens. If you look at the placenta, both bioengineered probiotic prevented Listeria from getting to the placenta. Are you with me so far? Because placenta, next phase is fetus, right? What about the fetus? We looked at the bacteria in the fetal liver, and so we didn't see much. And that's what we got excited that it worked. Of course, the brain, we didn't give enough time. So the brain, it takes more time to go through the fetal brain. So this data from fetal tissues, right? And so I'll show you the picture. Hopefully it's not going to bother you. This is naive mouse, the placenta, and they have a multiple fetus. Okay, and you have from 4 to 5 babies they carry. And so here, when you have a wild type probiotic LM, you can see all these fetus are dead, and you can see dark color. And if you have this bioengineered probiotic, in this case, we've been using actually Listeria inacua lap expressing bioengineer, and you can see this healthy fetus. And what about individual fetus level? This is naive. This is when it is infected by LM. You can see all bloody, and this color, look at this white, still natural color, and it's protected. So this gives you an idea that we are able to prevent feto-placental transmission of Listeria, right, from mother to fetus. So we'll be able to protect the fetus against listeriosis. So that's one of our drives, and this is our future goal. See if it can be used in the human application. And immunologically speaking, it did whatever is expected. Not to spend time. Also another study, I think I don't want to spend a lot of time, because I think we are very close to be done. And Nick also did that colitis model, and he showed real interesting results, and it shows the protection against the colitis. Again, you have a high level of HSP60 expression, and you can use this probiotic to prevent colitis. It works very well, and a lot of his data he showed. Again, like I said, in the interest of time, I don't want to spend a lot of time, but if you have a colitis or inflammation, and this probiotic is also going to work because of the similar mechanism. Okay? All right, so you can see that serum FD4 level went down because it repaired the leaky gut very well. So this is going to be a fantastic probiotic against any kind of leaky gut. That's what you're looking at. HSP60 expression also went down. You can clearly see here. All those other cases, you have a high level of HSP60 expression. And I want to show this video. So when you have this colitis, and if you feed these animals with this probiotic, you have about 89%, 90% survival rate. Whereas this DSS, which is the chemical-induced colitis, and it causes quite a bit of mortality. Let me show you this video. I think it will tell you the whole story. This animal, after they are treated with the DSS, you have a severe diarrhea, sodium feces, and then they started drinking this probiotic, right? You want to see them, they repair, they heal, right? And so those animals, actually this animal, this one, you see this one? Let me turn on the video. Let's see if it is, actually video is on. This animal is totally sitting there, not moving at all. And he had been on probiotic only day 4. He just recently received the probiotic. But it was in the dying stage, death stage. Let's look at the next one. So it's been already 6 days on the probiotic, that bioengineered probiotic. You can see this start moving, right? What about the next one? It's been on 8 days on probiotic. Same animal. It's over. We are looking at the progressively how they repair. And look at the one after 10 days of feeding probiotic. So you can see those people suffer from colitis or inflammatory bowel disease or severe damage. This probiotic can help because it binds to the receptor and then can make cell-to-cell contact and have a strong immunomodulatory response. We've got tons of data. We don't want to spend time, but showing that it works. We did microbiome also. I want to wrap it up. So every story I told you, this is my last slide, summary slide, saying that why and how it works. So essentially, if you have lactobacillus expressing these proteins, LAP, it can bind to the receptor. Okay, as soon as it makes the contact, whether you have a listeriosis or inflammatory bowel disease, it starts the magic, right? If it is competing with the Listeria monocytogenes, it prevents. So what you are seeing actually, it improves epithelial barrier function, right? So through myosin light chain kinase activity, pathogen opens it, and probiotic it closes. This is the opposite action. So that's why it is working very well. Anti-inflammatory response, all the cytokines, TNF alpha went down, IL-6 went down, and interferon gamma up, IL-10 up, TGF beta up. We want them. And then T regulatory function, Treg cell, and we saw this improvement significant. So it does immunomodulation. This is how it works. Okay. So we are not the only one doing bioengineering of probiotics. A lot of these are done for specific pathogens. They are using kind of like a vaccine carrier. This is a very good system. A lot of work being done, or still people doing a lot of work. I want to leave you with a story from a lady. She went through pregnancy-related complications. She's from Indiana. Her name is Amanda Wright, and she keep writing me letters because she saw our research. She lost her first pregnancy due to listeria. Okay, and she's trying to be mother for the very first time, and first pregnancy went through complications, and she lost the fetus. And so once she lost it, then she saw our paper. We published this, and of course we had news release, and saying that if you have a listeriosis problem, and a lot of times mothers suffer from that, and she was totally devastated because she lost her baby. Then she said, well, I want to be mother, so second pregnancy. That's when she started writing me. She's asking me, can I have this probiotic? I said, no, you can't, because we don't have approval. It needs to be approved before I can give it to anybody. And she was so convinced, so she kept writing, and so she started writing several letters. So during her pregnancy, I was kind of like I never seen her. She just writes me. It's like a pen pal. And so I'm kind of like a counselor, trying to give her advice. She's afraid of eating any food. Think about it. If you are afraid of eating food, how are you going to survive? You're scared that, oh, listeria may be there in the dirt. Listeria may be there in the water. Listeria may be in the leafy green or fruit or salad or sandwich I'm eating. You can be eating a nightmare. And she went through that second pregnancy, and fortunately she was lucky. She had a healthy baby. Then, as I'm going to tell her story a little bit because she kept writing, and these are some of those language she used in her email. Just trying to emphasize, a lot of times we try to address one person at a time. This is how I see it, right? If I can help one person. So she said, Arun, I have a crippling fear of listeria during pregnancy. Recently miscarried, and I'm trying again to conceive. So that was the second time when she started writing. And she wrote a long letter. I just gave some highlights. The work you have done is so important and would truly save so many lives, not just from the actual infection itself, but also in terms of the mental anguish. Like I said, any food she wants to touch, she gets so scared. And mental anguish associated with worrying about listeria. I really appreciate your time and hopeful for response. Anyway, then she wrote also another letter. She wrote, I would pay an arm and leg for this kind of peace of mind in pregnancy. And you can see this is a, like I said, I never met her, I never seen her, and I know she's from Indiana, that's the state I'm from, and she keep writing. Even I received another letter 2 days ago when I'm here, and she wrote me another letter. And she asked me very specific questions about, again, some food, and she's trying to have another baby. So this would be actually second baby. So she's going through pregnancy. And she said she doesn't buy any food from anybody. She doesn't trust anybody's food. She would prepare herself. And she asked me a very specific question. Because my internet problem, time to time, when I have time, I want to respond. I didn't get a chance to respond to her. I'm going to respond to her with that specific question. But I just want to leave you with this thought that the listeria is a huge problem. A lot of things happens here. You hear some of the stories. Sometimes doctors may not know. They cannot link that to this. I won't say that India doesn't have that problem. It's surely there, but we are not looking for it. In the Western world, we have it. We recognize it. We take care of it because we know the problem. It is there. And so that's how we try to help them. And so this probiotic works well because of the pregnancy-related complication. We showed that in the guinea pig model. At some point in future, some point that we want to commercialize to a way that it needs to have a FDA approval because it's a GMO, right? Genetically modified organism. But you can see, at least the animal study, it can save life, right? So what we are going to weigh out, GMO versus saving life. At some point, we have to approve this. So we are very hopeful. So we are starting with the animal model first, animal application, like a livestock. If they can improve their health, we know we can have approval. And then later on in the human application, especially like listeriosis, we would be able to do it. These are some of the recent people, new people. We had two interns came from NDRI. They just started and I left. They're supposed to arrive there by April. It was so delayed. So they are, these two are very active. And then these are other students in my lab. And we have a small group, but the group is very active. Usually a lot of this work I presented, some of the students work for 5 to 6 years to complete some of the studies. And it's pretty rigorous. I want them to start one hypothesis, you complete it, before that you don't leave. So essentially when they finish, they get rewarded with the nice papers, articles, usually high impact articles. And the job they get is really, all of them are professors or some medical colleges, they work there. So they have a really bright future. So anyway, I will end it here. I know I went a little bit over time because of the length of materials I wanted to cover. But the whole idea is that I wanted to give you a flavor of type of research we do, even those probiotics we're talking about, pathogens, biofilms. And you can see this, all of those applications actually exist in this research. Right? Okay, I will end it here if you have any questions. Come on, you should have a lot of questions, right?
So my question is, let's suppose we have a biofilm in dormant stage. So sir, can the prebiotics detect it or not?
You are saying the biofilm in the
Dormant state.
And the biofilm is made by actually the probiotic. That's what you're saying?
Yes sir.
Okay, so who is going to the immune system?
Yes sir.
Oh yeah, yeah. So they are forming it. So what we are advantage of probiotic forming biofilm, and you know that not only in the mucus, they are coming in contact with the epithelial cells. Are you following me? So when they're in contact with the epithelial cells, they can send the signal much stronger through the epithelial cells. And that signal would be resulted to the immunomodulatory response or the junctional response, right? So those signaling cascade would work much better. Even though they are in the dormant stage, they can still do that. And that's what we are looking for, biofilm formation. So I was trying to show you the data that these engineered bacteria they actually attach to the epithelial cells, and they actually form a large colonized biofilm-like structure. And that's why we were so excited, because once you form that, attach that, form that biofilm-like structure, you don't have to feed the animal every day. We did an experiment in that paper, you can follow it, that we stopped feeding bacteria or the probiotic, then up to a week and a half to 2 weeks, you can see the colonized probiotic. You see this? But if you have a regular wild type probiotic, they don't stay there. So they're in the mucus, and then eventually they go away. So that's why most of the probiotic people take it daily. Okay? But this one you don't have to take it daily because it has that property of forming biofilm. That's a good question. Yeah, if the biofilm, then some cells would be dormant, some would be active, right? And that come in contact with the epithelial cell. That's the key, epithelial cell contact.
Thank you, sir.
What do you have?
Hello. Yes, sir. Today's lecture was one of the best. Sir,
Okay,
my
great.
question is, many of the probiotics have these adhesion factors, the genes like MUB genes, sort of things, which help them to adhere. So, is there someone who is exploring that whether these genes can be used through CRISPR? Can they be used to enhance their adhesion factors in other probiotics?
That's wonderful idea. Essentially, yes, those are the approach we have to take. I don't know, I haven't read any paper along that line, but idea-wise, that makes sense. That would work very well. So some of the probiotics has the surface layer protein, right? They also make that contact. But the question is it reliable? Is it going past through the mucus to make a contact with the epithelial cell? This is the problem. The reason our system works because lab that has a strong affinity towards the HSP60. And so if there is an affinity, so if you even the bacteria on the mucus, they can pass through the mucus layer. Even they can pass through the strongly adhered mucus layer. Remember that? Loosely attached and strongly attached. Even we are showing that they go close to the epithelial cell. So even though natural probiotic, if they have these adhesion factors, a lot of times their affinity may not be well understood or strong enough to pass through the mucus layer. They need to go past the mucus layer. They need to get to the epithelial cell. That's where most of the probiotics don't work because they are way up there in the mucus layer, in the cloud, right? And they are producing the short chain fatty acids or all those molecules that diffuse through the mucus, then get to the epithelial cell. And sometimes because of the dynamic system in the gut, not all the times they are reliable. They are going to always find the positive attributes. But in our case, always I wanted to have the bacteria attached to the epithelial cell. That way you can initiate that signaling cascade and positive attributes what you saw, all those downstream effects. If you can make them attached, no matter how you do that, and that's when you can get the positive effect what we saw. Simple probiotic, we turn them into magic. You see this Lactobacillus casei. Now no secret to that. That wild type didn't do protect that animal. Did you see that? Didn't do anything. Those animals are dying. Wild type has no effect. So magic, happened because you put that protein and you took that from Listeria inacua. Now it's working very well. I know long way I answered your question. But that answer, okay. Any other questions? Come on, we learned a lot of stuff in last 10 days, right? Or I will not say 10 days, but at least 6 days, 7 days. Okay, I know a lot of times you don't want to ask in public. I know I'll talk to you in private. That's fine.